right, let's wait just a second for everyone to join. Hello, everyone. We'll get started in just a minute. We just need to get everyone to join. Hang on. All right. I think we're good to get started. So welcome everyone to one more of our FLUXNet Early Career Network seminar series. We have a really interesting, have a couple of really interesting talks for you today. And Christian Vishner, Gabriela Shirky, and myself will be helping with the questions on the background. And here's your host for today, Fred Wotulabi. Take it away, Fred.
All right, thank you. Hello, everybody, and welcome. Depending on where you are, good morning, or good afternoon, or good evening, and welcome. As Gilberto already said, we've got two very interesting talks coming up in the next 1 hour or so. By way of schedule, we have Karina Williams and Silvano Fares going to do the seminar for us today. Karina will speak first for about 20 minutes, after which we will take a few questions. And then Silvano will also come in for another 20 or so minutes. And then at the end, we'll have a general discussion around what we've heard. So without wasting any more time, I'd like to introduce our first speaker, Dr. Karina Williams from the UK Met Office. Karina leads the soil moisture stress work with the UK's land surface model called JULES. And I guess she would tell us more about her work in a talk titled Using Fluxes to Evaluate and Improve Land Surface Models. Karina.
Thank you very much. I'm just going to share my screen. Bear with me a second. Great. So hopefully you can see the slides now. Right. Thank you so much for the invitation. I'm really happy to be here talking to you on this topic. So I'll be chatting a bit about using flux measurements to evaluate and improve land surface models. And I was also asked to share a little bit about my career path so far. And I didn't start in land surface modelling. I actually started in another area of science, in particle physics. So for me, there's this common thread of modelling, but what I've actually modelled has changed quite a bit. And I think this is quite a common path into modelling, that people do arrive from lots of different backgrounds. And that has advantages. You've got lots of people with different perspectives. It also means that a lot of people, me especially, that don't have that in detail knowledge of actually what I'm modelling in real life, which I'm going to talk a bit more about in this talk. So I work on the JULES land surface model. So JULES stands for Joint UK Land Environment Simulator. And it's a land surface model. So it models water, energy, and carbon fluxes between the land and the atmosphere. And it can be used on its own. So it can be driven with observed rainfall and temperature and other meteorological data, or it can be coupled to an atmosphere model and driven with that. So it's the land surface model that's within the unified model that gets used in the Met Office for making weather predictions for the UK and other areas. And it also gets used in climate projections as well. So it's a land surface component of the UK Earth system model. So obviously, FLUXNet is this amazing resource to use for validating land surface models. You've got data taken from such a wide variety of biomes, and it can really test the model is behaving properly across the world and different timescales and different situations. So this is really important that the land surface modelling community use this to its full potential. And there are lots of ways that it's used within the community. So there are lots of tools which incorporate FLUXNet observations as well as other observational data sets, so satellite data sets or other data sets. And the example I've got here is ILAB, which you can find out more information about at ilab.org. And it's very good for comparing lots and lots of different models or configurations of the same model against a wide variety of different observations. So I've taken a screen grab here, but it's only the top bit of a web page. It actually extends really far down the page and uses loads of different observational data sets. And I just made sure that you could see a couple of examples of where FLUXNet observations are used directly here. So this is from the ILAB website, and this particular page is comparing lots of different CMIP5 models and CMIP6 models. So the models are written along the top there, and they're given a score. So red is not so good and blue is better. So that's a sort of overall score. So you can see a lot of models and a lot of different variables all in the same place like that. Or you can dig a little bit deeper. You can change what kind of metric you're looking at. You can change what region you're looking at. And for the FLUXNet sites, you can also map out the individual sites and how well compilations of models or individual models are doing compared to those observations at lots of different timescales. So there's this huge wealth of information here about how models are performing en masse in a way that's very easy to access and compare to each other in these sorts of tools. And that is absolutely invaluable. In this talk, I'm going to focus on something slightly different. So I'm going to be talking about how these site observations are able to really let us pull apart bits of the model and understand it, and understand how well that bit of the model does compared to another bit of the model, or if we want to improve something, really isolate it for improvement. So I'm going to be focusing on water stress within the model, in particular the effect of low levels of soil moisture on the vegetation. So low soil moisture affects the canopy assimilation and stigmatic conductance to water vapour, which means that when the amount of water in the soil does get low, this is a really important moderator of the fluxes between the land surface and the atmosphere. And this has a really big impact on the rest of the land surface scheme, but also on the coupling to the atmosphere model as well. And it really matters if you're looking at questions of, for example, how much carbon your land surface is storing and the effect that has on the concentrations of carbon dioxide in the atmosphere. If your model allows different types of vegetation depending on what the conditions are, so if your model will simulate changing vegetation cover as we go forward into the future, that's also going to be impacted by how much water is available to the plants and how they react to that amount of water. It will also impact how much carbon plants can assimilate to use for food. So it will affect crop yield and questions of food security. And because it's affecting these fluxes, it is having this impact on the local weather and climate from the feedbacks with the atmosphere as well. So this is one process that is quite key for a lot of different applications of a land surface model. And 4 years ago, within the JULES community, there were quite a lot of people that were saying, oh, actually, this process is not really performing as well as we would like it for the applications that we're looking at. This is one of the limiting factors in being able to apply our land surface model to as many different areas as we want. So there was actually a group that was set up, and it was across the JULES community, and it was trying to take in a lot of different expertise from different areas. So expertise from looking at actually how plants respond to different conditions, but also the other things that might affect that, like how the soil behaves and actually how the hydrology in the soil is modelled, so you know how much water the plant might have. And so it was really a cross-community effort within the JULES modelling community. So just to give a quick overview of this part of the code, just to make sense of some of the slides in the future, and there's quite a lot of commonality between JULES and other land surface models in this. In JULES, the effect of soil moisture stress is parameterized by this factor, which we call beta, which is 0 when the vegetation is completely stressed, and 1 when the vegetation is doing absolutely fine. There's no water stress, there's no effect of water stress on its fluxes. So this factor enters into the leaf photosynthesis. So it scales the leaf photosynthesis, and then because the leaf photosynthesis goes directly into the stomatal conductance calculation, it also means that stomatal conductance is scaled by this factor. So you can see that the leaf processes are very influenced by this factor. But then when you scale up the leaf processes to the rest of the canopy, that also means that you can see clearly that in situations where beta is between 0 and 1, so partially stressed, it can really have a big impact on the fluxes that you're seeing. And within JULES, there's also options for scaling respiration as well by beta. So the original way that beta was represented in JULES actually comes from a paper from 1998 that uses data from the Fife experiment. So the Fife experiment, I always have to look this up, stands for the first international satellite land surface climatology project field experiment. And this is data from 1987. And on the top plot here, this is the canopy conductance to water, and then this is the net canopy carbon assimilation in the bottom right. And using that data, this paper came up with a beta that was 0 below a certain volumetric soil moisture, 1 above a higher soil moisture, and linear in between. And those two limits were set to be minus 1.5 megapascals soil matrix potential and minus 0.033 megapascals. And you can see from these plots that it did really, really well at fitting this data. So this site is a tallgrass prairie site in Kansas. And during 1987, there was a dry spell in the summer. So this really gave the model a chance to look at whether it could reproduce this dry spell. So this way that beta is parameterized here, it comes from this one experiment. And then in JULES, because JULES has a soil hydrology scheme, so there's 4 different soil layers in JULES, and the fluxes of water between each soil layer is modelled. So JULES has to make a decision about how to weight the stress from each individual soil layer. So the default way that JULES does this is it looks at the proportion of roots in each layer and weights the beta that you would calculate from each layer according to that. And there's also another option in which you can define a root zone and then look at the average properties within that root zone and then calculate your beta from that. So that's how this stress factor is incorporated into JULES. And the group that I was talking about that's looking at evaluating and improving these processes recommended that actually the onset of stress in Joule should be delayed until lower soil moistures, which means effectively changing this beta. And this came out of a wide variety of different studies. A lot of them were using site data, and I'm going to give some examples in a minute, but it was also looking at online runs where you've got the land surface coupled to the atmosphere, and global runs at different timescales and for different applications. But uniformly, there was this recommendation that this function needed to change. So this diagram shows how it should change. The solid blue line here is the default line that we saw before. And the light grey lines here are actually lines taken from potted experiments where they grew a plant in a pot with lots of water and a pot with not much water and compared the difference. And by adding in an extra parameter into Joules, so this is it was just called P0, and this line shows it being set to 0.6. So this is, that means that is 60% of the total difference between the wilting soil moisture and the critical soil moisture here, where the default parameterization went to 1. So I'm going to talk a little bit about changing this value of P0 based on the site observations. So the first example that I'm going to show is Irrigated Maze from a site in Nebraska in the US. So this is FLUXNet site US-NE1. And on the right here, I've got the FLUXNet observations for gross primary productivity over the course of the year. And the coloured lines are the modelled lines from Joules. So the default line is the solid yellow line. And you can see actually it's a much wider peak, but also it doesn't get as high as the observations. And if we plot the value of gross primary productivity before the factor of beta is applied in the model, it becomes much higher. So we can see there's actually quite a big effect of water stress in this irrigated site. And as you imagine, this site shouldn't be water stressed. It's irrigated so that it doesn't have water stress. So this is not great. Because there's extra observations available from this site. So by working with the site investigators who sent us leaf area index observations and soil moisture observations, we can do Joules runs where those characteristics are actually set rather than calculated internally within the model. So the solid green line is what happens if we give Joules particular soil moistures as was observed at the site. And you can see that there's a slight difference. The peak is maybe higher here, but it's still not helping much. But when we use the observations for leaf area index, we can see that actually the peak now becomes the right width. And this is because within this default version of Joules, there's no proper representation of crops. So what we're seeing here is the gross productivity of effectively a field of grass. So by forcing it with the actual leaf area index, that means that what the model is now getting is information about when there's no leaves, so the crop hasn't actually started growing yet, and then the crop grows and it grows to a much higher leaf area index than natural grass would. And then it starts to senesce, the green leaf area index decreases again. And basically the seasonal cycle is far better because of this difference with leaf area index. If we look at that run before the water stress is applied, it actually really performs very well compared to the observations. So this is a very good indication that the water stress is overrepresented at this irrigated site, which we're able to pin down because there is so much information available at this site, and because we've been able to talk with the site investigators at that site who are able to give input on our runs and give us extra information that we can incorporate into our runs. So I'm just going to show an example of what happens in the model when P0 is increased. So the top row here is the gross primary productivity again. So that's P0 of 0, that was the default version of Joules. And this is P0 of 0.4. So the onset of water stress has been delayed a bit. The bottom row is the water fluxes, so the latent heat energy. And again, with P0 of 0 and P0 of 0.4. So we've got the observations in black and the model in green. And you can see that by reducing the water stress at this site, you do increase gross primary productivity much more towards the observations, which is the right direction. So we might even want to increase P0 even more at this site. And you can also see from the water fluxes that this seasonal cycle isn't quite captured. So given that we are now being able to pin down from extra information from the site, all the different characteristics, like we know that the soil hydrology is not relevant because we're forcing with observed soil moisture. We know that the phenology scheme in Joules is not affecting this because we're forcing with observed leaf area index. So that means that we can pin down the fact that actually this problem here where Joules is overrepresenting the latent heat flux in the early part of the season is not to do with the way that the vegetation is responding to water. We can pin that down to being another process entirely. It's the way that soil evaporation is being modelled in Joules. So by having that extra information, having that conversation, we're able to really isolate this process within Joules and also avoid misinterpreting the data. So for example, this is a picture that David Scobie sent us from the site. When he's been fantastic at giving us advice on how to use these observations and giving us all the different extra supplementary information that is available about this site. And this is 2010, this is after a particularly bad hailstorm, which is obviously going to affect the results. Is having this extra information is really, really useful. So given that the original parameterization works so well for the Fife experiment, it's worth going back to that data and seeing whether if we delay the onset of water stress, do we cause problems there as well? And actually there's loads of extra observations that are available for that data set as well. And the soil moisture observations and soil properties, really wide variety of different data sets that were taken by different teams with different instruments. And there was also leaf potential observations, which showed that actually in the early season, so May, June, the grass was mostly unstressed. And this is a plot of the soil moisture stress factor. The blue and the red is for P00. So this is the default onset of water stress. Whereas the green one is when water stress is delayed to slightly lower soil moistures. So P0 is 0.3. And if you're wanting to say that in June, the vegetation isn't really stressed, this is about as low a P0 as you can get away with. You can just about still count that as not being very stressed. Maybe you want it even higher again. So it was really interesting to see that actually there was evidence that this particular original experiment that was so influential on water stress within the Joules land surface model that would indicate that this threshold should be lower. And because there was lots of observations of leaf properties at this site, we were able to recalibrate the unstressed leaf properties to take into account this change in the water stress factor. This data was also very important for diurnal cycles. And this is one particular day during the dry spell in the Fife experiment. And the default Joules model is blue here. if you try and tune to all the available observations at that site, then you get something that looks like this green line. In both cases, though, it's forced by observed leaf area index and it's forced by observed soil moisture. So you're eliminating problems from the soil hydrology and from the leaf phenology scheme. And the crosses are derived from flux observations at that site. And you can see that actually there's this big dip during the middle of the day, during this dry day, in net canopy assimilation that's really not represented by the model. But as we saw, the original paper that looked at this water stress parameterization, it fit the data really, really well. And the reason that was the temperature dependence within the model. So it's a temperature dependence in terms of the leaf assimilation. And I hope you can see the line here. There's a red dotted line. I'm just moving over it with the hand there. This is the temperature dependence that was implemented in that original study. And it starts to decrease here at really quite moderate temperatures. And these are temperatures that are being experienced during the middle of the day here, which meant that the net canopy assimilation decreased nicely during the middle of the day from temperature. Whereas if you look at the observations at that site, which are the green crosses here, it doesn't support such an early decline. It supports a much, much later decline, which is why you don't see this dip in the green line here. And the default Joules parameterization is this blue line, which also doesn't start decreasing until much higher temperatures. So this is really key. The earlier experiment was able to capture this diurnal cycle because of the wrong process. It was a response to temperature. And actually, looking at the other observations in this site and the leaf potential observations, it's more to do with vapor pressure deficit in the middle of the day, which, as you can imagine, these sorts of things can have a real impact on how much you trust the model in the future when you're pushing the model into different regimes under climate change. So you really have to trust that processes are working well. And this rich data sets like this are really key in getting that confidence that your model is performing well. So another example is the dry season in the Eastern Amazon. So this is a FLUXNet site, BRSA1, and the data here is from the LBA, the large scale biosphere atmosphere experiment in Amazonia. And this is a site that on a typical year is not really water stressed during the dry season. There's quite a seasonal cycle at this site in rainfall, but the vegetation there is not really water stressed during the dry season. And so you can see the observations in blue. Actually, the gross primary productivity in the dry season, which is November, December, January, actually increased a bit, if anything, although it does stay remarkably constant. And default Joules, unfortunately, gets really stressed at this site in the dry season. So gross primary productivity just like dips like this hugely, which, I mean, that has a massive effect on the fluxes in that region in that particular season. And you can see that this is due to water stress, because if we plot again gross primary productivity before water stress is applied, it's this dotted line here. So the difference between this dotted line and the solid line here is entirely water stress. So the model is getting stressed where it shouldn't be getting stressed. So if we look at a different value of P0 for this site, it helps a bit, but it doesn't solve the problem. What really solves the problem is changing the soil column in Joules. So as I mentioned before, we have 4 soil layers in Joules. But those 4 soil layers only go down 3 meters. And at this site, it's just not enough to store the water that you need to keep the vegetation going through the dry season. This is an example from another site in the similar region. So the rainfall's on the top box, and then soil moisture is in the bottom box. And you can see that during the dry season, the soil moisture in lots of different layers of the soil well below 3 meters really gets affected. So having a model that only models the first 3 meters is just really not good enough for this site. So this is another example where knowledge about actually what is happening really helps pin down how we should be improving the model.
Karina, we've got just about 2 minutes.
Excellent. So the final example was looking at comparing different models, and we saw comparing lots of models on a very wide scale that basically model the whole world, all these system models, but also FLUXNet observations are really useful for detailed model comparisons of different processes. So I just wanted to flag up this evaluation of different maze models that look specifically at how they represent evapotranspiration. And by working with people who are providing the data and looking at what they think of the models and what processes should be improved, this really helps understand how maze should be modelled in general. So I have discussed a bit about how flux measurements are so important for evaluating land surface models, and that land surface models have lots of competing processes. And really pulling that apart is how you work out how to change a process, especially when it's something like water stress, where you have this extra factor that can just be used to compensate for loads of different biases. And this really works best when people doing the modelling work together with the people that have taken the data, that know the site, such that you don't end up having something that's some bias in the model compensating with another bias. Like, it's really down to those conversations to show how things should be improved rather than just giving an overall score to something. So what I was really hoping from the discussion would be that we could maybe talk about how this works at the moment. So the FLUXNet Early Career Network is fantastic, and it is such a good way to bring together lots of people from different communities all across the world that are working on these areas. But I just wanted to hear from you a bit about whether you have any other ideas of how these links can be strengthened, and anything perhaps I can take back to the modelling community that I'm in, and how we can work at strengthening these links to really make sure that the data is used to its full potential. Right, thank you very much.
Thank you. Thank you very much for that very interesting talk. We can have a few questions for Karina. If you have any questions, please type them in the chat, and then we would put them to Karina whilst we prepare for Silvano's presentation. There's one question here for you, Karina. So somebody is asking, uncertainty is a key part of models, and we are now seeing uncertainty estimates as part of measurements as well, like in uncertainties from flux measurements. How do you see these two parts of uncertainty estimates playing together, as in flux and JULES uncertainties?
A really good question. I think there's a few different ways that would happen. One way is if you have an idea of the uncertainty in your observations, then, and where it comes from, then it means, and you also have an idea of how influential that particular process is in the model, then you can prioritize which are the most important uncertainties. So by having that conversation, it can then direct research to the places that would, it helps you distinguish between which uncertainties are the most important ones to try and spend more time or get more data to pin them down a bit. Sorry, that was a bit convoluted. So it helps direct what observations you need to reduce that uncertainty if you know that it's particularly important for an application that you're looking at. But also, if you know that there's uncertainty, you can do model runs with different values of that parameter and explore how, and then rather than presenting just one number for your model output, you can present a range of model output. So it can, there is intrinsic uncertainty that can't be reduced, that has to be communicated in the way that you're presenting the model output as well.
Right, thank you. There are a couple more questions, but I would leave them for the discussion later when Silvano is done. So we will now move on to Silvano Fares, who is a director at the National Research Council in Italy. He's not new to the FLUXNet community. He's a member of the FLUXNet community. He's both into measurements and also modeling, and he would be talking to us about from flux measurements to multi-layer canopy model for ecosystem services in urban areas. Silvano. You need to unmute yourself, Silvano.
Okay, sorry. Thanks for inviting us, Fred. And can you see the transition presentation or the right one, first of all?
We're seeing the transition now, Silvano.
Is transition? Okay, let me change it.
Yeah.
All right. Okay, thanks then again. Okay, I'm talking to you about my experience in the last about 12 years. First as a PhD student in Italy, afterwards after postdoc in Berkeley with Allen Goldstein, and with the strong collaboration also with Dennis Baldocchi group. And in the following 10 years as a PI of a flux measurement site in central Italy named Castelporziano. Let's start from some very, very simple introductory slides that can somehow help focus on what is my presentation is about. For me, when I talk about exchange processes, I'm focusing on three major compartments in the canopy. First are the stomata. The stomata are open and regulate carbon, but also non-carbon trace gases, and in this and DOC, volatile organic compounds. So stomata is the major sink. But we also have the surface deposition of cuticles and soils. And another sink, which I believe is very fascinating to study, is the chemistry in the gas phase. So reaction in the canopy space between the DOC and ozone, for instance, but also with many other trace gases. So there are these three major sinks of gases in the canopy. Given that not for 100% of people, it's clear that when I talk about ozone, I'm talking about tropospheric ozone, the bad one, the one that is detrimental for people, but also for plants when it enters through the stomata. So my research is also addressed at trying to understand what is the effect of ozone when it penetrates to the plants. So I'm talking about the tropospheric ozone, not the stratospheric one, just to make it clear. And something else I wanted to clarify is what VOC are. Not everybody is an expert in studying these things. And so VOC are trace gases produced in plants, and the biosynthetic pathway of production is very much connected to the photosynthetic pathway. And why they are produced in very tiny amount. You can find VOC in the air at the level of very few parts per billion. And so they play a role in the defense process against pathogens. When the VOC volatilize in the airspace, in the canopy, in the ecosystem, they somehow act as an hormone, as a phytohormone, so they affect other plants and organisms. They favor the ecosystem perturbation. They get to the atmosphere and they are involved in a lot of photochemical processes. So they play a role also in the air quality. And there are a bunch of hundreds of VOC The most common is isoprene. It's a hemiterpenoid made of 5 carbon atoms. Then there are bigger VOC in terms of structure, like the monoterpenes, which are 10 carbon atoms. And then there are sesquiterpenes. They are very, very reactive VOC made of 15 or more carbon atoms. They are very emitted in a very tiny amount, but when they are emitted, they react very quickly in terms of few seconds. And just to give you an idea, when you walk into the natural forest, the majority of VOC which are in the air are biogenic. But in the city, VOC, of course, are also anthropogenic source, non-vehicular traffic, combustion processes. But still, you can trace biogenic VOC in the city at the ratio 1:1 with anthropogenic. So the VOC signature in the globe is pretty much biogenic. Again, we are talking about fluxes, eddy covariance. So my interest is to study fluxes not only of CO2, of course, CO2 is my major gas because it connects to the ecophysiological property of plants and the performance of photosynthetic machinery. But I'm also interested in studying fluxes of trace gases. Once you have very precise and fast measurements, we have technology nowadays to study VOC fluxes, ozone fluxes, NOx fluxes. And when we do CO2 fluxes, we can easily get to an understanding of the gross primary productivity, which is something that is pretty much close to the photosynthesis at the level of the canopy. When you do water flux measurements with some interesting analysis, you can also derive the stomatal conductance at the canopy level by the inversion of the Penman-Monte, so solving the energy balance in the canopy. And when you have stomatal conductance, you can do many things like estimating stomatal ozone fluxes, so that portion of ozone which penetrate to the stomata, which is so important to determine the effect of ozone on the plant physiology. Okay, so what I'm showing you some case studies coming from 3 experimental sites where I've been working on my career. The first one is Castel Porziano. It's an urban forest. It's a holm oak forest nearby Rome. So the advantage of this place is that we can somehow measure and test the effects of anthropogenic emissions coming straight by advection phenomena just on the field site. So we have this holm oak contaminated with air quality compromised by the anthropogenic activity of the city of Rome. But similarly, we can also, well, just a slide to show this is a site, of course, which is a Mediterranean site, which is exposed to somehow the drought stress. And we see very abrupt increase of GPP when we have some wet years. So we are measuring GPP since 2012, and we have seen that, of course, this site is drought prone, and of course, it's the ecophysiological features are very much related to the precipitation regime. And the second site where I show you data is a citrus orchard, so it's an agricultural site, which is in the Central Valley of California. It's one of the most polluted areas in the world in terms of say ozone exposure, for instance, because it's between Los Angeles and San Francisco, convergence of a lot of polluted plumes, and stagnation of air masses. So this is a perfect site if you want to study the effect of air pollution on the physiology of plants. So for 2 years when I was in Berkeley, we have been doing flux measurements at this site. And the third site is the famous Blodgett forest. This forest provided data for 7 years on the row. It's a Pinus ponderosa forest on the Sierra Nevada mountains. Again, another site which has somehow Mediterranean-like climate and is very much exposed to pollution coming from Sacramento and San Francisco. So 3 sites very well defined for their characteristics in terms of exposure to pollution. Let's start with the flux description. Okay, I was mentioning about ozone. Ozone is never produced by ecosystems, it's only sequestrated by ecosystems. And so there is a strong deposition all year long. We can quantify this with eddy covariance. 8 gram ozone meter square, it seems like very low amount, but consider that ozone is present in terms of few ppb in the air. So it's quite interesting sink in terms of the ozone canopy. And as I was mentioning, we are able to somehow estimate stomatal conductance from eddy covariance measurements of water vapor. We apply the Penman-Monte equation, basically inverted them among the equation. If you don't know this formula, somehow that formula which somehow estimate the evaporative demand of the atmosphere based on the resistance that the water can find from the canopy to the atmosphere. If you already measure the evapotranspiration, you can basically invert the formula and estimate the stomatal resistance. So with this trick, we get an idea of what could be the stomatal conductance. And the stomatal conductance can be used to calculate the stomatal flux, because you just multiply by the concentration of ozone, which is just at the entrance of the stomata that we measure with sensors. And again, with this method and with other, let's say, atmospheric models, some of them are very recent, some other are very old, we can somehow get an idea of how ozone flux in the canopy is partitioned. So this is a first indication. We measure the total ozone flux via eddy covariance, and then we can individually estimate the single component of ozone sinks into the canopy. If we are very good in this modeling exercise, we can basically explain where ozone goes to the canopy. What we have found so far that in most ecosystems, the majority of sinks is stomata. And we confirm this by estimation of models. This is what concerns the site in Castel Porziano, but also we have seen similar ratio in the citrus orchard, where we have incredibly high ozone level, very high. It's like these are daily average that sometimes exceed 100 ppb, very phytotoxic thresholds. And same in the Pinus ponderosa forest, in the Blodgett forest, where we have about 30 to 60% of total ozone deposition into the canopy due to the stomata. Again, a very interesting correlation between the oxidation products of ozone at the Blodgett forest and the non-stomatal fraction of ozone fluxes. This is already telling us something, that perhaps some of the ozone which goes into this canopy is somehow removed by the VOC. So we estimate that up to 40% of these sites could be an ozone flux due to the VOC. This is very interesting. We can interpret VOC as a defense mechanism in a certain way, because the ozone doesn't really get into the stomata. It reacts outside the stomata in the canopy space environment. And this is some experience from the Castel Porziano site. This is VOC fluxes. So far we have been talking about ozone, now we move to the VOC. And this is the typical daily dynamic in the emission of VOC. VOC are emitted in response to light and high temperature. So we expect high fluxes during the central hour of the day. These are monoterpenes and isoprene. And an interesting phenomenon is observed when you measure just the concentration of VOC in the canopy. You will see that from day to night, concentration change very much, in part because of the boundary layer. And this is something clear. But also we see an accumulation, for instance, of very reactive sesquiterpenoids during night hours, for instance, because sesquiterpenes are emitted in response to temperature as well. So even at night, there is no ozone at night, and is titrated during the day. So you have accumulation of sesquiterpenes. During the day, you expand the boundary layer, so you will have lower concentration, but at the same time, also ozone builds up and react with sesquiterpenes. And so we will see a total decay of sesquiterpenes. We don't see them anymore. At that time, you will have an increase of non-stomatal ozone fluxes because of this reaction with the VOC and also with the stomatal penetration. At the same time, you see a concentration product of oxidation of isoprene, which is methylketone and metacrolane, which is the green line in this graph. So, and later on during the day, you will see oxidation product of acetone, which is another VOC emitted from vegetation. So just studying the concentration can get a lot of ideas of what's going on into the canopy. And this is what we measure in the Central Valley of California, very interesting fluxes, so a small range of VOC, but already you have an idea of what's going on. Flowering event, blossom of flowers, a lot of smell, a lot of VOC emitted from flowers, high fluxes. Then you have harvesting, trimming, so titration of leaves, heavy working with the material, so a lot of release of VOC, and you see high fluxes. Application of fertilizer, again, high fluxes. So you can see during the course of the year, the intervention of the site generate these high fluxes. And so coming back to the modeling exercise and how good job we did in the Central Valley to estimate the total fluxes by comparing with the measured eddy covariance ozone fluxes. We did a good job outside the flowering season. We didn't do a good job during the flowering season because perhaps the rule of VOC, basically we underestimated the rule of VOC because we were not able to measure all of them. If you can see the bottom left graph, is a measurement with a time of flight PTRMS, a very sophisticated instrument that gives us a potential to measure a broad range of VOC, although even if we are not able to classify the species of VOC that are there, there are thousands of them. So there are some phenomena that we still don't know exactly the complex blend of VOC emitted into the atmosphere. It's very complex air quality dynamics. Okay, so I'll go to a second part of my talk. I don't know if I already abused of my time, maybe a few minutes more. How to use eddy covariant fluxes to test and validate canopy models. Well, this is just an example. It's called IR3. It's a simple multi-layer canopy model which resolves the energy balance into different layers of the canopy. There is also an hydrological model to simulate what happens in the soil, and it works at the canopy, at the ecosystem scale. Something that I developed in my lab, but it's very much similar to many traditional canopy models that we know. And validating the model with eddy covariance data is something that I think is extremely, tremendously important. And so what happens is that in a sensitivity test of this model, I realized that the most sensitive parameters are soil property, humidity, just to find correlation with the previous talk, but also with the ecophysiology of plants, like the velocity of carboxylation of the rubisco enzyme, for instance. So the model is extremely sensitive to this. If we don't parameterize very well the model with these two parameters, you will have wrong estimation of dynamics. This is an example of GPP. This is an example of measurement of the velocity of carboxylation with the portable instrument measuring gas exchange at the leaf level during the year. You can see a curve, but also in the canopy profile. So the ability of a good canopy model would be also to interpret the different values at the level of different layers. This is very interesting. This is an example of how the model simulates the partitioning of ozone fluxes in different layers, somehow depending on the amount of foliage and the biomass in each layer, but also the energy balance closure of each layer. Interesting, the model doesn't make a good job when we have high emission of VOC. It's not able to yet to predict relevant removal of ozone by the VOC. And this is just an example of how to apply model at a different scale. This is a peri-urban park in Rome, and we use IR3 to estimate the carbon removal and also the particulate matter and ozone deposition on this huge park after we validated the model with eddy covariance measurements. This is just an example of introduction of MEGAN, which is the model of emission of gases and aerosol from nature, into IR3. MEGAN is the most worldwide used empirical model. It uses empirical algorithm predicting VOC emission in response of light and temperature. And when we use MEGAN, of course, and also we simulate drought stress, we were able to observe some changes in the VOC emission in response to drought stress. At a different scale, this is thanks to a visit I did at the NCAR in Colorado a few years ago. We were able to use the eddy covariance flux measurements of VOC at Castel Forziano and estimate the basal emission factor. What is the basal emission factor? It's the emission potential of plants at a given light and temperature regime. This is a fundamental number to go into this model because of this number, you can basically estimate the emission at the level of the entire region. So what I did at the NCAR is just to run MEGAN, which was incorporated in the community land model, basically, imposing the basal emission factor I measured in the field, which was quite divergent from the basal emission factor of the general plant types that was into the database of MEGAN. And we saw relevant changes also in the air quality and in the ozone concentration prediction because of these little changes of basal emission factors. For instance, the old oak forest is a strong monoterpene emitter. It's not an isoprene emitter. Like the oaks in the United States are strong isoprene emitters. So the Megan, which was developed in the US, foresees that let's say, the oak ecosystems produce a lot of isoprene, which produce a lot of ozone. In the case of the Mediterranean Europe, it's the opposite way around. The oak forests produce a lot of monoterpenes, which don't generate that much ozone, but perhaps they produce more fine aerosol. So you can see how important it is to parameterize this model with the data that we measure into the field, especially with the ecovariance. I just jump on these other details. In the very last topic of my talk, I want to mention if we can estimate the ozone damage to the vegetation using the ecovariance measurements. This is the most challenging part of my work because, as you can see from this graph, there is a correlation between stomatal ozone flux and GPP. Makes sense, right? Because the photosynthesis is correlated to stomatal conductance, which is correlated to the ozone entering. And the correlation of stomatal ozone fluxes works much better than the total ozone flux measured with ecovariance. But also there is a lot of noise because there are many covariates in this. So you can see the R square quite low. Another thing you can see is that if you go to study a database of data, and let's say a time series of data, sorry, you may identify some periods in which you have high ozone exposure, and period where you have low ozone exposure. And what we see is that the slope between stomatal conductance and GPP slow down at episode of high ozone concentration. It looks like ozone has an effect on stomatal aperture. This is something we can see from the field. Another see that if you go in the frequency domain, we can study the temporal correlation between GPP and ozone stomatal flux. And somehow the temporal correlation is not that strong when the ozone concentration in the air was very high. It seems like the high ozone concentration broke the temporal correlation between GPP and ozone. And if you apply some more conventional statistics, we can build linear and non-linear regression models. For instance, this is a simple stepwise regression where we have basically estimated if the presence of stomatal ozone fluxes into a GPP model could somehow play a role, and indeed it does. We can also apply non-linear regression, like the random forestry analysis. Even in this case, when we use several predictors, one of them is the stomatal ozone fluxes. It has a part in estimating GPP. For instance, we calculated that during high ozone episode, we could have a GPP loss up to 20%, which is a big number in very polluted area, of course. And last, we could also use neural network analysis, which is a very much conservative approach because only returns us up to 2% of ozone damage. But this is another method that we are exploring in the last few months. And this is my last slide. Okay, now we are challenging the idea of using ecovariance data to estimate ozone damage. So far, only control experiments are being used to do this. And what we can do is also we can build refining new metrics for ozone risk assessment. What is a metrics for ozone risk assessment? It's something that tells us that after a certain exposure to ozone, you reach a critical level where you have a loss in productivity up to 10 to 15%. Okay, why don't we build this metrics with ecovariance data, with real-time data in the field, rather than using, like, for instance, open-top chamber or manipulative experiments? This is interesting. So why don't we tune multi-layer canopy model, we validate them with ecovariance, and we train this multi-layer model to predict ozone losses? This is our ultimate goal. And what we can do, we can use the conventional algorithms which are in the multi-layer model, like the Ball, Woodruff, and Berle algorithm, and apply a stress factor. We can also apply a stress factor to stomatal conductance, okay? And when we apply this stress factor, we compare the model output with the ecovariance, especially in episodes where there is no ozone stress, for instance. What we can find is that we may perhaps have an estimation which is more realistic when we incorporate a stress reduction factor for photosynthesis and stomatal conductance. This is what we found for Castelporziano and Blodgett, for instance. We also found that the Castelporziano forest is very ozone resistant because its Mediterranean sclerophyll leaves very tight with a lot of waxes. So we expect less ozone damage in there. Indeed, to have a damage, we need a threshold of ozone exposure, which is very high. It's called 3 to 5 nanomole meter square per hour. In vice versa, Pinus ponderosa is very well-known ozone-sensitive species, and the threshold is lower. So introducing a lower threshold, we have a better agreement between model output and eddy covariance data. Again, this is in my opinion, a very promising field. We should invest more, even because we have a network of these sites. In Europe, we have ICOS, which is building a lot of new infrastructure of flux measuring in the long time. You have NEON in the United States. There is the big umbrella of FLUXNet community. So in the future, I expect we use more and more of this data. So I hope I didn't confuse your mind with so many data. And I want to leave the word to Fred. Maybe there is some time for questions. I don't know. Thanks a lot for.
Thank you very much for that very interesting talk. I've got one question for you before we'll see if we can do a bit more discussion. Since eddy covariance fluxes have limitations and VC max measurements are hard to scale up, are there any other strategies that can be used for such validation work in models?
That's a good question. I mean, we have realized that if we try to change too many factors, sometimes we made more mistakes than just not changing at all. I mean, what I'm saying is that VC max ideally should be measured on the canopy profile, but if we don't have this, we should use a number for plant functional type. That's it. I don't have that many other way of estimating this part. I think this is the weakest point of many models, especially those who work for, of course, for a regional scale, the VC max. This is one of the more sensitive parameters.
All right, thank you. Any more questions for either Silvano or Karina, or any general comments? Okay, there's one question here. Would you recommend threshold studies conducted at flux sites to help with water stress and ozone? How can we help parameterize? And this will go to both of you. You can take turns at this.
Who starts? Karina, go ahead.
Sorry, can you repeat the question?
So the question is, would you recommend threshold studies conducted at flux sites to help with water stress and water and ozone stress? And how can we help to parameterize these?
Threshold studies for threshold for when water stress is important, or generally any studies that probe water stress, I think, is very important for our model and a lot of other models in the community. Silvano?
Yeah, I got kind of fascinated by those super sites which are now being built in many places in the world. There is one in France, for instance, in a whole mock forest, for instance, where they do flux measurements with eddy covariance, but also do manipulation of water stress regimes with some sheltering of the bottom of the. So, I mean, this would be step very, very nice to test exactly. Because the problem about the covariance is that we have too many covariates, and we struggle with trying to exclude the effect of covariates, but the methods we use are not that precise. So if we can isolate a stressor like the drought, that would be a relevant enhancement in our knowledge approaches.
All right, thank you. Dennis, I can see your hand up. Do you want to unmute yourself and then ask your question or make your comments?
Hi, Silvano. Very nice talk. I guess the question we all ask is, what's the availability of reliable ozone sensors? In the past, they were all kind of homemade. And has there been any improvement that we could have sensors that we can operate year-round, like a CO2 and water vapor sensor?
Thanks, Dennis. This is a very good question. I'm in Europe. We are discussing about this because we are trying to somehow see if we can implement ICOS protocol with some additional sensor to be used. So far, we have been using a lot of customized models, and the one I've been using until last month is still a one that NOAA customized for me when I was in Berkeley 12 years ago. So the answer is no. I am not aware of very precise sensor, which is very well known. There are a couple of companies, one in Australia and one in Austria, which are producing these instruments and are selling these. And they all work with the same principle. They have coumarin dyes. And so to a certain way, they are very precise because they have this photomultiplier, which is very, very sensitive to small variation of ozone. But of course, there is not that instrument that I would suggest to use because it's very well known, like could be, for instance, a Lycor product for CO2 or a Picaro for methane. Yeah, to my knowledge, there is not that one sensor that is the best one. But sorry, let me add that, for instance, the concentration measure on the other way around can be very precise because we use ultraviolet sensor, like for instance, Thermoscientific and other products. They are very reliable. And for many studies, you just have to measure ozone concentration. You don't need to do anti-covariance measurement of ozone. For instance, to measure stomatal ozone fluxes. If you have a very well-equipped site for evapotranspiration measurements, then you also couple with an ozone concentration measurements. It's totally fine. I mean, it would be great if all sites could have just measurements of concentration, not fluxes.
I think we still have time for a question or two or a comment. If you have any, you can raise your hand and then unmute yourself and then just go ahead.
Hello. Can you hear me?
Yes.
I know it's just on the ozone sensors. I mean, that's something that I've spent quite a lot of time working on as well. Which I think Dennis and Silvano and I have talked about it before. But no, there's still not a commercial instrument that's readily available that you could just use like a Lycor, unfortunately. I mean, we've been looking at the ecophysics, the titration-based instrument, but we haven't really been able to get it working at a level yet that we would run it in the field continuously. So it's still an area that I think we should, needs a bit of development, but the technology just doesn't seem to have come up yet that's going to do the job, unless anybody can tell me of anything new that's come out.
Yeah, a few years ago with Mairi and other colleagues, we made a field campaign in northern Italy, in Bosco Fontana, where we deployed all kinds of ozone sensors, and it took us days of intercalibration. And that was the most challenging part, to make every sensor read the same value. After that, I think we found out that these coumarin-based sensors are not that bad. Of course, they are not that reliable in the long term. You have to change these coumarin dyes very frequently, and it would be very wise to have a standardized protocol that is adopted worldwide when you measure ozone fluxes, because the decay of the sensitivity based on this coumarin is quite high, and depending on the frequency of maintenance, and your sensitivity depends on that. Your precision depends on that.
Mm-hmm. Yeah, I mean, the coumarin-based ones are nice, and in some respects they're simple and easy to run, but we still haven't quite got the. I've been working on a methodology to try and standardize it a bit, but the paper's still a work in progress. So
look forward to read it.
All right. If we don't have any more questions, I'll go back to our speakers for their last words, starting with Karina.
I guess I just want to say thanks again for inviting me. It's been really great to be part of this. Thank you. If you do have ideas of how anything we can do from our side to strengthen collaboration, please email me and we can discuss them further.
Thank you. Thank you very much for coming and for giving us such a good talk. Silvano, any last words?
No, the only thing I want to say that I look forward to see all the community in person at the next meeting, whatever is EGU, AGU, but I have enough of staying home. I think it's a common feeling for everybody. I am very happy to see some faces in this list of people, very good friends, and that I don't see in months or years. So I really look forward that we're gonna meet soon at the next meeting, maybe the next FLUXNet meeting or the next ILAB conference we are having in starting 2022. Thanks.
Okay. Thank you very much, everybody, for coming, and we hope to see you again at the next webinar, which we will announce and email around to inform you. Thank you very much, and see you next time.
Bye.
Bye-bye.
Thanks. Bye.
That was a really nice talk, everyone.